I am excited to welcome everyone to the second interview in our new Lab to Literature series brought to you by the American Society of Microbiology Subcommittee for the Status of Women in Microbiology. I'm Amy J. Reese, the chair of the subcommittee and an associate professor at the University of Health Sciences and Pharmacy in St. Louis, Missouri. It is my pleasure to introduce and to lead the interview today of Dr. Katalin Karikó, who won the Nobel Prize in Physiology or Medicine with Drew Weissman in 2023 for their mRNA work that was foundational in the development of the Pfizer, BioNTech, and Moderna COVID-19 mRNA vaccines. Dr. Karikó is a Hungarian-American biochemist who has worked passionately to follow her curiosity to understand RNA-mediated mechanisms for over many decades. She was persistent when the scientific community was skeptical of its value, when she didn't receive funding or get promoted, and when her positions were cut. Through that all, she just wanted to learn and pursue excellent science. And she did, and she continues to do so. Dr. Karikó, or Kati, has recently published her memoir, Breaking Through: My Life in Science with Crown Publishing. I first got a copy of it at a display table at a meeting for first-year experiences as it was recommended for first-year reading for college students. I've enjoyed reading it twice and encourage you to pick it up after our discussion today if you haven't read it before, and it's an excellent read for anyone interested in science, the pursuit of science, and the balance of life and science. I have some questions that we will use as we get started, but we welcome questions in the audience, in the question and answer or chat at any point. But first, we want to start with Dr. Karikó. Would you please share with us the highlights you want us to know about you, your journey, your career, and your book? Thank you.
Thank you, Amy. That was a very nice introduction. Yes, I would like to say a couple of things about my life, and I thought that I would just present it as um, just in shortly describing that what is all about what happened. And so I I born in Hungary and I grew up in a very small town, Kisújszalás, 10,000 people live there. My parents had only elementary school education. My father was butcher, my mother was bookkeeper, and I learned from them that hard life, the hard work is part of life. And of course, how to make sausage, because we learned this also with my sister, older sister. And so I have to say that nobody would learn about my life unless 5 years ago, the Boston Globe had an article when the COVID-19 vaccine was approved, and then they mention about my life, the interesting life which I have here. There are a lot of ups and downs. Like at age 30, I lost my job in Hungary. I get in the U.S. a new job. Then I lost in the US in the new job. I get a faculty position at age 33 at Penn. Then I was demoted from the faculty. Then I did working and do major discoveries, but again, I lost my job at Penn. And then I went to Germany, and at age 58, and 10 years later, I get a Nobel Prize. So immediately that was like an interesting life. And then, they want to explore more. I have to say, I competed in elementary school. My sister was studying very well. Later, she got a PhD in economy, but I was interested in science. And I was in chemistry and biology competition, and I recently I found a book, which I started to write at age 13 about animals, and I made all of these drawings. Please do not look at too close to this crab. I had too many legs there, but maybe I was just very enthusiastic little girl. And at age 13, because I was competing in biology, was participating in biology competition, so I was in a camp, a biology camp. Here I am, the tallest one. But many of these girls, I later met them in life, became chemists, biologists, or here is Susan, she is a physician. So it is very important that even age 13, kids are knowing what they want. And what happened is I didn't know why I was writing about animals, the book, because I actually love plants and early on discovered that, in addition, I had this illustrated book, and every time I found a plant, I tried to find the name. And then I discovered that there is a Latin name and there is an L after the name, and that is because carbon lineage. So at age 14, I wrote an essay about Linné because, I realized how smart he was, that every things had to have a common name, a Latin name, so everybody would understand. And 55 years forward, when I get the Nobel Prize, I was in University of Uppsala, and I they presented me the book what Linné was writing. It is similar to my little book I started at age 13. He was 24 when he was doing these drawings, so I was not too bad after all. Anyway, I went to the high school and we sent letters to famous scientists because famous Hungarian scientists because we couldn't speak English. We didn't learn English there. And for me, it was important that we sent Shea a letter and he responded. And then, we read his book, which was published in Hungarian. And at age 16, I learned how to handle stress. And I wouldn't sit here, I wouldn't write any book, I wouldn't get any award if I won't learn early age that how to handle stress. Because that helped me, that I don't blame others, I don't complain, and I don't carry grudge against anybody. And we learn and practice there in school. And after all, the main message is if you want to do something, you find a way. If not, you find excuses. So think about that when you find excuses. And in addition to mental health, the physical health is also important. So I was always exercising, even when I was in Germany, 60 something years old, before going to the lab. I run 6 kilometers next to the Rhine River. So I learned in this all of these things in high school that you have to have a plan, whether you writing an essay or so you finish it, you work on it, submit, and that's it. Of course, we know that the reality is not that easy because there are always problems and the goals getting further away. But we have to learn and how to get out those stitches. And one thing is to start with right when we are young to get out of the comfort zone. And then we can learn and experience. And not just learning more about the new environment, but we learn so much about ourselves. So everybody can decide, we born and now today we are here in our lives. At the beginning, the decision was made by our parents, but later life, we made decisions. And this is in the future also, that which way we will start and going. And from Shaya, we learn, who actually coined the word stress in the 1930s, that, when we make a decision, we always had to look forward, what next, and not agonizing on that. Oh, if I would have gone in a different direction, then it would be different. He said that never, because it's never worry about this, because it's very stressful. So don't worry about it. You made a decision, so what next? So I still was in high school when for underprivileged children were organized a camp. So in the middle of the summer, for 2 weeks, we could study, and it was helping those whose parents were not highly educated, and helped us to prepare to get to the university. So we couldn't get the pass and get to the university lower score. We had to have the high score, but we get opportunity for that. And here I am on the right on this picture here. It was from a newspaper. Anyway, I get to this university because the biological center was just open, and I dream about one day I will work here, working with plants. So as a student, I went there, but there were no opening, only in the lipid lab. As my father was butcher, I knew that lipids are important, but I didn't want to work with that. But I imagine I working in the bench here, but the supervisor sent me to field trip, and he told me that can spend the rest of my life, working at the bench, but here I am, go out in the pond and collect some water fleas. At that point, of course, I didn't know that at age 58, I still pouring the gel and culturing cells and other things I would do with my own hands. Anyway, so as a result of it, this was my first publication, came out with working with this little water fleas. But I dig myself into, the research and with the lipids. And when I return in the fall to the laboratory, two scientists were interested in to make liposomes. And then for that one, they needed certain lipid fraction, and they came to the lipid lab to isolate that. So again, the head of this team, he knew that if you want to do something, you find a way, because in 42, this was published, how to isolate these things. We were behind the iron curtain, everything was embargo, we couldn't purchase things. And so we isolated those things, those lipids, and we did transfer DNA into mammalian cells. And one day, the head of the RNA lab came and asked me that whether I want to work with RNA. So I was not a visionary person every time. I just went with the flow. I started my work to make this RNA molecule, but the head of the laboratory, he went for sabbatical for 3 years to the United States. So I worked together with a fellow student. So this can happen. Everything was very good. We were progressing, but I was terminated in my position. So meantime, I had a family, I married, and our little daughter was born, and now at 95, 40 years ago, we ended up in the US. We just purchased this Pinto, this car. We didn't know that how dangerous it is at that time. Anyway, so going to America was important because the information, 85, there were no PubMed, never knew, no YouTube and many other things, no internet. So the only things I felt at that time the most important for me to coming America is that I could go to lectures and listen and learn what others are doing. And so I ended up in Temple University and working with Professor Suhadolnyik. And later, after uh, removing from his lab, I ended up in the University of Pennsylvania working with Elliot on different projects. From 89, I worked with RNA, and we did project that he was interested. And I had difficulties to get funding because when you do research, you have result, and then you publish, and then um, uh, after publication, you write grants because you expect to get money and increase the team size. But I was, shortcut here and always get back to research and more generating more result and publication. But others get bigger team, and they get promotion and appointment, and somehow it start the career is number one for the team head, and the students had to get their PhD, and then they publish when there's not much to say. So I say that sometimes this collecting scientific knowledge and advancing career is not overlapping. Why the goal of the research should be understand nature, and that was in the 19th century. Scientists, they try to understand nature, but in these days the scientists want to publish in nature and not understand it as much. Anyway, I started to work after 7 years in cardiology. 14 years I work in this lab in the corner here you can see, thanks to David Langer, who I met him in Elliot's lab in cardiology, and he was neurosurgery. And he convinced the chairman that neurosurgery need a molecular biologist. So I did all of this work at this bench. I worked 17 years. No much funding, so no lab coat or safety gog or other things. But I was very happy. and doing all of the research. And one day I met Drew Weissman at the Xerox machine, and we discovered that RNA is immunogenic, inflammatory, but changing one component of the RNA still translate very well, but is not inflammatory anymore. So that was our discovery. And everything was good. We filed a patent and we established a company, RNARX, and this is the only grant I ever get. This $1 million is small business grant. Everything was good. My daughter grew up and then she got a Olympic gold medal, one in Beijing in the women eight, US women eight, and then she got in 2012 one in London. But again, I lost my job. I was terminated and forced to retire from University of Pennsylvania. And because I felt that I still have so much to offer, so I ended up in Germany, not in this nice building, but in BioNTech was in the campus. And I can say that was in 2013, and 10 years later, there are 6,000 people work for this company. We did a lot of work together with Özlem and Ugur, and when it was 2023, I received the Nobel Prize. Everybody who was important was there. My husband, we just celebrated 45 years. We are married, and my daughter, my sister was there, and those who I did with the lipid research, and my teachers were there from Hungary, the chemist and genetic teacher, and of course Elliot and David Langer, who helped me at Penn, and those young ones from uh, BioNTech. And in these days, books were written about me, what I was doing. So just inspired the kids here with Drew Weissman. We are doing all of this research. And I wrote this book with the help of Amy Benjamin. And then it was published in many other languages. Uh, on Saturday was also out in Iran in Persian language. And so this is my story, and let's talk now about more about the book.
Thank you so much. And I hope for the audience who might not have been as familiar with your story or your book, where you also share some of those images, really get a chance to get a better feel for you. The first time I read your book, I was really struck by the story when you joined the lipid lab and you needed to make ethyl acetate for this first time on that job. The second time I read your book, I had to read that passage out loud to my daughter because your persistence really stood out. And you shared a whole list of excuses that you could have employed, but that you didn't. And I wondered if you could share a little bit more about that story with our audience, who might not have read the book. And how you perceive, we use excuses so often, but how you just didn't. And if you could share that with us.
Yes, so as I mentioned, I was sent out to field trip, and there was supposed to be a supervisor there. And the first day when I arrived, he just said that he's going to vacation. And now I am here, a student, and I had to analyze lipids from the fish, which was fed with different show material, and I have to run gel apparat, and I have to run in a TLC, this thin layer chromatography. So I knew that how to make a thin layer, with the silica put on the glass, because we couldn't purchase anything, when I was working at the university. So I knew how to do those things. And then it was like different components I needed for this solvent that I had to put the plate in. And, I couldn't find the components. And I asked them that whether they have ethyl acetate. That was one component. And they said, oh, they don't. So, but at least they have ethanol. So I looked it up, in the Merck index, how to make ethyl acetate, what is the boiling points and how to. So I started to find apparatus that I can do it. But I learned that glacial acetic acid, which was needed, they don't have it. But they said that they can call up and I can go to the Rice Institute because there was an other research center in this small town. And so on the bike, I went over and get a 1 liter of glacial acetic acid. So then I get the two components, and then I start to assemble the glassware and then measure the temperature as I learned in school that how to distill and collected that fraction. And then I was already having one fraction for this solvent that I needed to separate the sample. So that was, also always comes there that, if you want to do something, you find a way. If not, you find excuses. And, I could complain that this guy, why he invites me here when, he's away and there were no components. And I could enjoy just the day sitting next to this lake and enjoy the time. But I want to work.
Yeah, the collection of ways that you shared, and this could have you could have had all these barriers, and yet you were like, no, I got to figure this out. I will be doing this. Nothing's standing in my way. You also share stories about a teacher along the way that you realized uh, really wasn't rooting for you, and how you compartmentalize that. And I think we all experience that. And I wonder if you share
Mm.
how you address that.
Yes, so one of my teacher he didn't like me at all, and so when I was a valedictorian and everything, I just finished the exam and came out. Everything was fine. They praised me, and but he came out and he said, "I know somebody at the university. I make sure that they will not accept you." And by that time, I was 18. So I 2 years practicing how to handle stress. So I didn't punch him on his face. I didn't say anything. But I made the decision that I will study all summer. I will be the number one. They have to accept me. And this is the decision because I could decide that, okay, I don't even go to the exam because he already knows somebody and will arrange it. So, but this is also the reason why every time I get an award, I say thank you to those people who, try to make my life difficult and miserable. Because if he would say, Kati, I know somebody, I will talk to him, make sure that you will accept it. Isn't that I would study less? So thanks to them, okay, I studied more and I learned more. So they made me work harder and, I learned more. So thanks to them. But this is the thing that what is in the children would decide that which way to go, whether, when, let's say, just they have a running competition and they are number one in the class, and then the teacher said, okay, go to the district competition, and he comes out last. And that decided whether I don't come again running because I don't like to lose, I don't like to be the last one, or whether it says I will practice and come back next year, I will be the one, number one. I have to clone that gene and turn it on in the children.
Thank you. And you do also share in the book something you just alluded to now, that the compassion that you have and the thanks that you gave to the folks who did make it more difficult. You made choices to thank some of the folks you worked with who made it harder for you, and you chose to be gracious about that. And I wonder what lessons and advice you have for us about dealing with that in that moment and making that choice.
Indeed, so we learned from Shaya that you shouldn't have grudge against anybody and never think about revenge, because the most important things to learn and study is that you have to calm down. You have to have this, he called it homeostasis, so nothing is bothering you. The only thing you have to focus on your research or whatever. And the only way you can reach is that when you very angry with somebody, you have to think about why should I grateful to that same person. I was just ready to slash the tire or something because he was so horrible to me. And so that calms you down, and then it will be, not escalating, because if you do revenge, then it escalates and hits you back, and not good. So that like Professor Suhadolnik, who reported me for authorities. So I am a fugitive. I get a job offer. I just told him that I get a job offer and I would like to go to Johns Hopkins, but he called them up that I am a fugitive. They shouldn't hire me, and they withdrew the offer, and and he made my life, that I will not get a recommendation letter from him. And the only reason he did this because he wanted me to stay in the lab. And every time after he did these things, he called me back. Your bench is here. Come back. Because I worked very hard for 3 years. So not that I was bad and he wanted to get rid of me and wanted to deport me. And so, but when a couple of years later, I went back and he was in the audience, I was thankful, and because he invited me to the United States, and that's what I emphasize. I didn't say that almost he get me out, but and I learned so much in his laboratory, and that's what I emphasize when I told whatever I told during my talk there was true. So I was grateful, and I was grateful, to people that really treated me badly, but I appreciated also that the good thing about them.
Yeah, so focusing on those good things and what we can learn
Yes.
from that. And yeah, thank you. You also emphasize in your book strategies for success. And one of the things that's very relevant to this committee that's hosting for women in microbiology, you comment on what it takes for a woman to be a mother and a successful scientist. And you showed pictures of your daughter, and I wonder what you've gleaned from living in various places and your insights from this. And I wonder if you'd share with us about that balance and challenge.
Yeah, so for scientists to have a life work balance, family is not easy, and but I was lucky that in Hungary, when I gave birth to my daughter, there was a very high quality, affordable childcare. So every time I give a lecture, I always ask, is anybody here from the government to have this for the women to get affordable, high quality childcare? Because registered nurse were there every day. Pediatrician visited, so we could ask questions and they could check the child, and they get all of the vaccination there so I didn't have to take a day off to get the vaccine for my daughter. So but I also realized, recently they, like 2 weeks ago, they told me that it was cruel that I put my daughter in the daycare, because the child need the parents. But I have to say, when I was with her, it was quality time, because many people who are, stay at home, they have their telephone and they are just, okay, just, watch TV, and they are not really spending that time. At least, I and it was, I didn't give up my career, because then I would be bitter that I give up so much for, and it's not needed. You just have to have right partner with your husband, or help, and family will help you, with raising a child, and it is fun. And I have to say to the women that don't overdote your child, because you rob them the opportunity to learn how to fight for themselves, how to stand up when they fail. And so it is important lesson for them. And they will watch you, and if you work hard, they will try to imitate you.
Thank you. You also describe some strategies that your hometown looking out for each other, and then also the system of schooling that you experienced. You share in the book about a system of marking by student names with an F that makes something very different than, of course, what we see as an F in the United States. And you share about your education musings. What if we had a system like that? And I wonder if you'd share a little bit more for our audience about that process and how you think you might have benefited from it and how others would benefit from it.
So indeed, in Hungary, this was a communist system, and they put emphasis on that everybody should get education if they work hard and they are qualified. And so if somebody had physical workers, so work with their hands, like, my father was butcher, then, we get the letter F, which meant that we could we could attend extra afternoon program, summer program. Of course, those who were not F, they could also come, but we get this opportunity and go to, for example, the university, organize something for the middle of the summer. So not every kid would love 5 o'clock, get up 5 o'clock in the morning, middle of the summer for 2 weeks and attending physics and mathematics classes and others, but we had this opportunity. And also the teachers paid more attention. So if somebody was studying very well, and then let's say the parents were not educated and never thought about, like in our family, nobody had uh, uh, even high school diploma, that they would, if we finish high school, our parents would not think about, oh, we should go to university because we have a talent. But my teacher, biology teacher, told me when I was 16 years old that I could be a scientist. And it was written in the big book of the class. I just recently have seen that, and it was two that I want to be a biologist, and two biology teacher from that the high school was their name was written that they had to pay attention on this, that helped me on that. But probably if not physical work, they also had assistants, and they opened for all of these after schools program. They could come and, but it was paying extra attention of those, and because this education was free, so, we didn't have to pay anything. I even get stipend, I get the housing, the food, everything, as long as I keep a certain level of academic achievement. Because if we go under that, then, maybe I would be kicked out from the dormitory, or I had to pay for the food or something. So that incentives they give to those who couldn't afford things. And, that their parents wouldn't think about uh, to go to university. My father, I describe in the book, he asked, what you are doing, research. I would understand if you are a teacher, go there and teaching the kids, but research and go what you are doing, what you would. So it is would be, of course, my parents would never think about that I could be a scientist. And of course, I have never seen one, but because the teacher told me that I could be one, so I believed him.
Thank you. I want to turn some attention to the questions that we've gotten in the question and answer section, and I invite the audience to continue to contribute. I want to start with one. Can you please tell us about deciding to be an author for your autobiography? And I'll put the title in and again in a minute. Um, and was it difficult to write about your own life? So for those of you who haven't seen it, so here's the book, and we'll put it into the chat. You can tell I've got lots of tabs on reading mine. But breaking through my life in science, what inspired you to write the book, Kati, and to share with us?
Hey, Amy, I have to say that when the Boston Globe article appeared, then I get the request that I want to write your life story. I said, why people would do that? I told my daughter, maybe we have to write. Maybe you can write because you know my life and you know English and so on. And she said, oh, okay, I will take care of. And then she found the agency, and then the agency, found we could select somebody who would help me to write. And when I was writing, I constantly was thinking, who is interested in the boring life of a scientist? I'm just sitting there with pipetting, running a gel, how a average person would understand how happy you can be seeing a bend, a smudge, that, oh, that's it, it is difficult. And so I have to say, when the first 100 pages, I don't know how many times we rewrote after, with Amy was writing after discussion with her. And then we rewrote. I always felt that it is so boring. Nobody will read that. Yeah, but eventually, that I work and write and rewrite, and then I don't know how many times. Then it was had to cut 20 pages. Then we had to lawyers look at that. Who can sue? Who is dead already? Who I am talking about? Oh my God, this is so complicated.
And I see there are a few raised hands. It would be helpful if you put questions in the chat or in the question and answer period. Uh, so it sounds like it's a bit of a reluctance approach into to the writing process, but we're so glad that you did. A question just came in. You had a sense of well-being and generosity of spirit even when the going was rough. How does it feel to you now that you have a Nobel Prize and are very much in demand? Yes, that it was.
I was happy when nobody knew who I was. And I
I
have to
believe
say
that.
that it was so quiet. At the first in 2020, when December, CNN just asked me a live interview. I mean, we are not prepared as a scientist. I didn't even know what is my name. I was just so nervous and, important question I have to answer and I have to, conscious about everything. So it took for a while that I realized, okay, I get certain attention and award or something, but I am representing all of the scientists. All of those who I was 40 years, nobody gave any award, and I was fine with it. But, you know, everything what I accomplished is based on reading many articles, many people's contributed to my knowledge, not just those teachers and mentors that I had, but the people who published and I was reading their paper. So I realized that okay, when I get an award, the attention is on science, on a scientist, and we have to celebrate that because it rarely happens. They are talking about the Kardashian and who knows what they were doing, but that was the main news, and rarely they talk about scientists and science.
You have a question in here. When did you realize that your work on mRNA encapsulated vaccine would become a blockbuster? When did you know that it was going to hit the main stage?
In BioNTech, we already started to collaborate with Pfizer in 2018,
Yeah.
so 2 years before. And we already, at that point, we want to develop mRNA-based vaccine for influenza. And 2019, right before the pandemic happened, we already ready for to have the trial started, human trial. People don't know that prior to the pandemic, there were the rabies mRNA vaccine, the Zika mRNA vaccine, CMV, and some others already in human trial. People thought that this was the first time that mRNA-based vaccine was tested in humans. So it was not true. So we already have seen the data. It was, for the influenza, and it was very good. And we were I was very optimistic. So I was not really surprised. But of course, eventually I learned the end of the trial, the phase 3. It was November 8th, 2020.
Thank you. I have a question here from the audience. I suspect your life at times, based on what you have and haven't shared, was nothing but easy or clear. What advice do you have for a woman in academia that isn't clear on what direction she's headed or where her place might be in science? So those still kind of struggling to find their way.
It is what I would say, that it is important to do something which is important, because then you spend so much time, your life, with something, constantly thinking about that. It has to be important, that has a influence. And it is difficult sometimes, to get funding. It is always difficult. So I never get funding for example, from the NIH R01 grant, but at least I could convince a colleague that what I am doing, and I convince them that how important and how interesting it is. So Elliot Barnathan and David Langer were there to support me and stand there. So if you are on the periphery there is no prestige, no respect, but you have somebody connected with who has all of these things, the money and the influence, and then convince that person, then you can proceed with your work. Because you can work in academia. I was not very successful in academia because these references and publication number and these, and for me, if I do the experiment by myself, I cannot publish 5 papers in a week because I had to write, had to read. And so this is challenging. In industry, if you are a small company, then you feel every time that, the survival of the company depends on you. So that's what at the beginning at the BioNTech was, that everything is so important and we work like 24/7 because this is very important, and everybody work together. There is no competition. And
Mhm.
it is good because it's not first out or last out, or it is a product. And if we don't have a product which is treating somebody well, then there is everybody can go home. If you go for a larger company work, for example, it is more stable sometimes, yeah, that the position, but you might not see the whole picture. You are in a little part of the big machinery. But it depends on you. You can explore what is going on and you can contribute more. So everywhere, in academia, industry, there are advantages and some difficulties there. But um, there are so many diseases there that we have no treatment because we don't even understand that what is the disease, how could it be, and what causing it. So there are so many things to discover, and so it is the young one who are listening, they will be they are the next generation. They will do it. And so it is deciding that what is hot today, I don't know. With this RNA, I just went with the flow, and I could see that improving of my work constantly, could see better outcome, and so I went with it. Because people ask, when do you stop? Because you just don't give up for not giving up. But when you repeat, and you don't see any advancement and you don't see any progress, then you have to maybe change what you are doing.
Thank you. There's a question about maybe your thoughts on the potential for CRISPR-Cas-based technologies and future vaccines. So maybe your current thoughts on the vaccine process and development in general, or that one in particular.
I mean, Cas9 is clinics also, and we could hear like this year when Children's Hospital here in Philadelphia, this newborn was have this problem, the ammonia accumulated, and very quickly they could manage with the mRNA coding Cas9. They could help her, and um, and so it is. There are every discovery, whether it is RNA or Cas9, it is something that at the beginning they discover something, what's going on. Then scientists develop that, oh, that could be useful for that and that. And they made small companies testing out things. And when something is promising, then they move to large companies who are running clinical trials. So there are a future there, and what they try to do now is to deliver, let's say, the enzyme coding mRNA to the bone marrow, so that wouldn't be so expensive, let's say, treating sickle cell anemia, thalassemia, to take out the bone marrow, process it ex vivo, put it back, but just sending the messenger RNA to the proper cell types. And that's what, again, something very important field to work right now. The formulation, how to you put in a lipid nanoparticle and how to decorate the particle to send to certain type of cells. So that's a big challenge.
Yeah. What public policies for advancing science do you recommend for the world at this point? You've been focused on the mRNA, but what's our big public policy challenge right now globally? The question is about developing and developed countries, but I think it's a global question for us.
Oh, there are problems at every level. In the most advanced country, like here, that policy can distract people coming here, and they, in other countries may be more favorable and helping scientists to advance, and of course, in developing countries, they also have their challenge of maybe they don't have enough financial support. But I think that more and more institutions like University of Pennsylvania, I remember like 20 years ago, started global uh, health, and they accept scientists and applicants to get to the medical school from different developing countries. So when they return, they have the connection with somebody who stay in different more developed countries, that and the connection is there, the personal connection, because they were studied together. So it is more helpful. And because different organizations are there, but maybe there are not as much personal connection which could help them. But uh, also the like BioNTech was helping countries in Africa, giving them the technology and training them. And and of course, in China, you get a lot of opportunity. mRNA is advancing there. When I look at people ask me for clinical trial for different mRNA application, I can see always many of them is ongoing in China, and 150 clinical trial ongoing, more than that in the world, and different application. Most of them is for vaccines, for bacteria, or mostly viruses, but parasite like malaria. And I recently Sanofi started like acne, for the bacteria that causes um, acne. And there are many of them is cancer related. Some of them is cancer vaccine. Some of them, mRNA code for antibodies or cytokines, and many of them is rare disease. But I came just from a month ago. We had the mRNA health conference, and actually, mRNA using treatment for autoimmune disease. That was the spotlight. So there are a lot of application, and I don't know whether that was the question or that was the prior question, but that's you can find a position on this field many different places, and hopefully that helping developing countries, because the mRNA is very quickly can be made and it is cheap,
Thank you. I
equalizer.
want to, one of the things that you make very clear is how well read you are. And I think somewhere in your book, wasn't that you had just read 9,000 papers. Those are the ones that you'd cataloged. Those are the ones that were worth it to you. There were many. And so I want to use this question about how methods of sifting through the ever expanding number of papers and journals. How do you navigate the information of today's world? Because I know from reading your book, you want to learn everything you can from everybody you can. How do you make that work for you?
Yeah, so for me it was easier mRNA. There is one paper per year, so it was easy to follow, and so it was. When I was reading a paper, I usually, because I was so manually doing everything myself, so I always started at the technique. How did they? I know. Oh, that's not right. So I won't even read the paper, because I thought that, oh, that's misleading. But today is mostly, I set up alerts so that PubMed alerts, Google alerts, and so they are certain scientific groups or people, or topic. I can set it up, and then I can, end of the week, I can get all of the abstract and reading about. And because so many things is not reliable, so when I read a paper, I start to read, I get the second typo in the I already stopped reading because I said, or when I find something that in a review, which I know already that is not true, because later it was disproved, but I could feel that probably a student is writing that who doesn't know about, and then
Mm.
I stop reading it. And so it is difficult to navigate. Many times when I was coming up with a new idea and I realized that somebody must measure this, that must be known. And I start to write and read and finding reference within the reference, another reference. And finally, in the morning, my God, I didn't no time to sleep now. And because I had a feeling and get always new information, so it can be endless. Because when at the beginning, prior to 2002, I had to go to the library because from 2002 I get everything digitally. Then I can, search, but then, at least I could sleep because library open in the morning. But yeah, it is very challenging to know. And the papers at when I was uh, 40 years old, the papers were 5 pages, not 35, 50 pages in today's. So it is challenging. And what is interesting is in today's paper is so much data, and you get lost. What is the thinking? And the old papers in the 60s, 70s, 80s, you find more of what was the scientists were thinking and the data, some of them supporting that thinking. Because listen, how we do as scientists is we do an experiment and get some idea. Then we reading, and create a hypothesis. And then based on this hypothesis, we design experiment to turn it over or prove it, confirm it. And so you never do an experiment that, oh, let's see what happens. No, you always have something expect, a bench, a band, or something you have to expect. And of course, when you don't get that, then you start to think more and learn more. Always when things are not fit what you are expecting, that you learn most. Because when you, okay, that's it. And you don't even think all over. So be happy when you get something weird data.
Thank you. I think that is a lovely spot to move to our next piece. I want to thank you so much for sharing your stories, your insight, and your time with us. Audience, I invite you to continue to learn more about Dr. Karikó. Her book is very accessible. It's not written for other scientists. It's written for the lay public. So please, I encourage you. Um, we've been talking today about the writing process and the reading process, and some of you might have book ideas. And so we're going to hear now from ASM Press on how that process can happen.
Thank you, Amy. What an honor to hear Dr. Karikó. Really inspiring. And I want to thank the subcommittee on the status for women in microbiology for inviting me today. I have been asked to speak with you about the publishing process, the book publishing process at ASM. I think what I'm going to do is, as the acquisitions editor for the press, and I'm here to represent what our mission and goals are. But if you want to if you have a book idea and want to get in touch with me about those mission and goals, I would love to tell you more. Could you go to the second slide, please? Thanks. The one before, please. Thanks. So we'll skip over mission and timelines and I'll just get straight to, if you have a book idea, you can send it to us if you go to the QR code on the right, it'll take you to a link to submit your book idea. And if you're interested in learning more about the press, just go to the QR code, which will take you to the link for the ASM Press page. Our goal at the press, we keep our numbers really small in terms of what we're publishing. So we aim to publish 10 to 12 titles a year. Not publish, sign 10 to 12 titles a year. And we do that because we want to maintain our reputation for editorial rigor and quality of books that we publish, and also to give the author a personalized, sort of first-in-class book publishing experience. We offer a lot of support every step of the way. Right now, we're actively seeking, as it says here on the slide, we're seeking titles in clinical microbiology and applied microbiology, nutrition, bioinformatics. But please don't, if your book idea that you have doesn't meet one of these, please send it to us all the same. We love to pore over all of the ideas, and some of them may seem way out there and a little bit far afield to you, but some of those are our best book ideas that come in. Next page, please. Thanks. So the very first thing to do is to just get your idea to us. You can do it via that QR code, or you can just email me at the press. And we don't have any rigid rules on the format that we're looking for. Some publishers want a 50-page proposal laid out in a certain way. You can send me a sentence. You can send me a paragraph or 50 pages. And if I think that is a book idea that meets the needs of our members, the ASM members, then we'll hop on a call with you and help you flesh out a full book proposal. And by that, I mean sort of articulating the concept a lot more, identifying what type of book it is in terms of, is it a reference book? Is, a manual? Is it a textbook? Is it a how-to book or a case studies book? And who the target audience is. The ASM members range from academics to people in industry and working in the federal government to graduate students. And so we just want to know which of our target audience you're aiming for. We will take a look at competitors for you so that you and we understand it very deeply, what the competition is in that field. And then determining whether you want to edit or author a book. And I go over all of the pros and cons of doing one or the other. Once we have the book proposal put together, and I really work very closely with editors or authors in putting that proposal together, then we take it to our publishing partner at Wiley. Our publishing partner at Wiley distributes, warehouses, and markets our books. They have no say in the content of our books. That is exclusively between ASM Press and our authors and editors. So we bring the proposal at this point to our publishing partner at Wiley and ask them for concept approval, if you will, to go ahead and do a full book, a full evaluation of the proposal. And what we do at this point is we send it out for peer review. I work with you, the author or the editor, in identifying 5 to 8 experts in the field. And we share it with them. And when we get that feedback, we share that with you anonymously and work with you to update your proposal as needed. And then the ASM editorial staff, while you are working on reading through the reviews and seeing what needs, what's in scope and what's out of scope and how you're going to update the proposal, the editorial staff at ASM Press are working on completing a sales projections analysis, which includes predicting the sales of your book for the first 3 years and making suggestions on price. And we're very transparent in sharing all of that with you. And then we take all of that information, we take those reviews, we take your amended book proposal and the sales projections, and we then present that to Wiley, where we see at that point if they want to proceed with a contract. I won't spend too much time talking about the contract, but if we could go to the next slide, please, just in the interest of time. Please reach out to me if you do have an idea. I would love to talk with, I love talking with our members, and even if you're not a member and you have a book idea, I would love to talk with you about what happens after a book contract gets signed. We continue to work with you very, very closely throughout the entire process. So in a nutshell, that's the process of publishing a book. with ASM Press. And please, as I said, be in touch if you have a book idea that you'd like to share with us. And for now, thanks again to the subcommittee, and I will pass it back to Amy.
Thank you. I want to thank you all for joining us today. I give our immense thanks to Dr. Katalin Karikó for taking time with us to share with us, for helping us think about managing our own stress responses, to get out of our comfort zone, to pursue our passions and interests while connecting with others who both support us and maybe push us. And Kati, thank you for helping us understand nature and for leading the way. Thank you so much.
Thank you. Thank you for inviting me.
Thank you all. Please go get her book if you haven't. Look for future events like this coming from our committee.